Measuring EMF's impact on brain activity, a comprehensive review. Electromagnetic fields, EMF, have become an increasingly prevalent part of modern life, from the smartphones we carry to the power lines that energize our cities. As our exposure to these fields continues to grow, understanding their impact on human brain activity has become a critical area of scientific research. This article explores the methods, findings, and implications of measuring EMF's effects on neural function. Understanding EMF and the brain. The nature of electromagnetic fields. Electromagnetic fields are combinations of invisible electric and magnetic forces. They exist on a spectrum ranging from extremely low frequency, ELF, fields, such as those produced by power lines, to radio frequency, RF, fields emitted by wireless devices. Each type of EMF carries different amounts of energy and potentially interacts with biological tissue in distinct ways. The brain's electrical nature. The human brain operates through complex electrical signaling between neurons. This intrinsic electrical activity makes the brain potentially susceptible to external electromagnetic influences. Neural communication relies on precise timing and specific voltage changes, which could theoretically be disrupted or modified by external electromagnetic fields. Measurement methodologies. EEG, electroencephalography. EEG remains the primary tool for measuring immediate changes in brain activity during EMF exposure. This non-invasive technique uses electrodes placed on the scalp to detect electrical patterns in the brain, providing high temporal resolution of neural activity changes. The key measurements obtained through EEG include monitoring alterations in alpha waves operating at 8 to 13 Hz, changes in delta waves functioning at 1 to 4 Hz, patterns in gamma waves ranging from 30 to 100 Hz, and various event-related potentials, ERPs. Magnetoencephalography, MEG. MEG offers another non-invasive approach to measuring brain activity, specifically detecting magnetic fields produced by neural currents. This sophisticated technique provides several advantages over traditional methods, including higher spatial resolution than EEG, enhanced detection capabilities for deep brain activity, and significantly reduced signal distortion from the skull and scalp. Functional neuroimaging. Advanced imaging techniques provide crucial insights into EMF's effects on brain function through two primary methods. First, functional magnetic resonance imaging, FMRI, measures blood oxygen level dependent signals, reveals areas of increased or decreased brain activity, and provides excellent spatial resolution. Second, positron emission tomography, PET, tracks changes in glucose metabolism, identifies areas of altered brain activity, and proves particularly useful for longer-term exposure studies. Research findings. Acute effects. Cellular level changes. Studies have documented several immediate effects of EMF exposure on neural cells. Research has shown significant alterations in calcium ion flux across cell membranes, along with modified patterns of neurotransmitter release. Scientists have also observed changes in membrane potential and variations in action potential frequency. Cognitive performance. Research has revealed mixed results regarding EMF's impact on cognitive function. In terms of attention and reaction time, some studies demonstrate decreased reaction times during EMF exposure, while others indicate no significant changes in attention span. These results often vary based on the frequency and intensity of exposure. Regarding memory function, short-term memory appears more susceptible to EMF influence, while working memory tasks show variable results. The effects on long-term memory remain inconclusive. Long-term effects. Adaptation mechanisms. Extended exposure studies have revealed potential adaptive responses in the brain. These include the upregulation of protective cellular mechanisms and modified gene expression patterns. Researchers have also observed altered neurotransmitter receptor density and changes in neural network organization over time. Chronic exposure considerations. Long-term studies face several significant challenges. Researchers must contend with the difficulty of controlling for confounding environmental factors and the limited availability of longitudinal data. Additionally, ethical constraints on human exposure studies and the considerable variability in individual susceptibility complicate these investigations. Measurement challenges and limitations. Technical challenges. The field faces two primary technical obstacles. First, signal interference presents ongoing challenges, as EMF sources can interfere with measurement equipment, necessitating sophisticated shielding techniques and making it difficult to separate environmental from experimental EMF. Second, standardization issues persist due to varying experimental protocols, different exposure parameters, and inconsistent measurement methodologies. Biological variables. Individual differences play a crucial role in research outcomes, including age-related susceptibility variations, genetic factors affecting sensitivity, previous exposure history, and overall health status. Environmental factors further complicate measurements, as researchers must account for background EMF levels, temperature variations, time of day effects, and the psychological state of subjects. Implications and applications. Clinical applications. The therapeutic potential of EMF research extends to several areas, including the treatment of neurological disorders, pain management, depression therapy, and cognitive enhancement. Additionally, diagnostic tools have emerged from this research, enabling brain activity mapping, neural disorder identification, and treatment response monitoring. Safety considerations. Safety guidelines have been established at international levels, with specific occupational limits and special considerations for vulnerable populations. Protective measures have also been developed, including various shielding technologies, distance recommendations, and exposure duration limits. Future research directions. Methodological improvements. Advancing measurement techniques represents a crucial area of development, with researchers working on more sensitive sensors, better interference elimination methods, real-time monitoring capabilities, and improved spatial resolution. Standardization efforts continue to focus on establishing universal protocols, consistent reporting methods, and reproducible experimental designs. Emerging areas of investigation. Research into frequency-specific effects has become increasingly important, particularly regarding 5G and higher frequency impacts, differences between pulsed versus continuous exposure, and frequency-dependent biological responses. Population studies have also gained prominence, emphasizing long-term epidemiological research, vulnerable population identification, and occupational exposure effects. Conclusion. The measurement of EMF's impact on brain activity remains a complex and evolving field of study. While significant progress has been made in understanding these interactions, many questions remain unanswered. Continued research using increasingly sophisticated measurement techniques will be crucial for establishing definitive cause and effect relationships, developing appropriate safety guidelines, identifying potential therapeutic applications, and understanding long-term health implications. As technology continues to advance and EMF exposure becomes more prevalent, the importance of accurate measurement and thorough understanding of these effects becomes increasingly critical. Future research must focus on developing more precise measurement techniques while addressing the current limitations and challenges in the field.